I'd like to thank people for coming to our first meeting in February of GLIMPRINT and associated organizations. We have a talk today by Emilia Luca from Sunnybrook Research Institute. As always, I need to remind people that this meeting is live streamed on YouTube and an edited version of it is made available afterwards for use. Tom Helikar, myself, Jim Sluka, and Lorenzo are always available to take your suggestions and comments. We appreciate your ideas about how we can do things better. Don't hesitate to contact us with ideas about who to speak, what we can do better in the future, anything that you think would be useful for us to do. We have a variety of communication channels. They work better if you use them. In particular, we have our archive of recorded talks that should probably be more distributed. We'd appreciate your help getting the word out about that and about advertising the seminar series going forward. If you have a publication in this area that you would like to share, there is the IMAG webpage. We can put things up there. If you talk to Jim Sluka, he'll help you do that. There's also the GLIMPRINT page. You can list things there. We are working to improve that page and make it more functional. But if you have job announcements, grant funding ideas, seminars, conferences, we can announce them on that page as well. Coming up, we have quite a full schedule in February and March. We have Lisbeth Garris talking on the 12th with Maria. We have Philip Gerley on the 19th, February 26th, Andy Exelson. John Hickey is going to talk on March 12th, and Reid Chapman from NIH, who's one of the leading advocates for multiscale modeling in the area of immune and infectious disease, is going to speak on March 19th. So we have an exciting program coming up in the next few months. Please help us keep it going. Suggest people you'd like to hear from. And again, really do help us build an audience because the speakers deserve a good audience for their talks. We want to call attention to the EDITH roadmap for the human virtual twin, I should say European virtual human twin, because there are many different orders in which these things are said. Also call your attention to the upcoming virtual physiological human meeting, which will be in the fall in Milano. They have a deadline coming up for abstracts for that meeting. A lot of people there in immune modeling. We have our OpenVT reproducible agent-based multicellular modeling competition open till the end of February. If you or one of your students or friends has published a multiscale multicellular model in the past 2 years in a referee journal, please consider submitting to that. We made it easy to submit. It's a very simple process to submit. It takes less than half an hour. We would love to have a wide variety of models to consider. If there are any questions about that, email openvtprize@gmail.com, and we'll do our best to help walk you through the process of submission. We are not going to have a steering committee meeting tomorrow. If there are any other announcements before we get started, now is a good time to let us know. Anybody have anything that they want to bring up before we start? So today we have Emilia Luca from Sunnybrook Research Institute talking to us about the Utricle, a microcosm of human balance, and I will turn over the screen share to her.
While Emilia's setting up, I just want to encourage everybody to really listen to her work because she's trying to offer a collaboration with theorists, which is, we've always been looking for something like this. And it's not just a collaboration, there's also a postdoc. So she's open to theory collaboration. So see where you can fit in, or if you would like to have a chat with her afterward, this is a real open call.
Good morning, everyone, and thank you, everyone, for the introduction. I'm Emilia Luca, Research Associate in the WWP Lab. I would like to thank the Virtual Human Development Consortium and the Bio and the GLIMPRINT organizers for the opportunity to present our recent published work and upcoming projects, and thank all of you for your interest in my talk. This is a human utricle, one of the 5 balance organs in the inner ear. You rarely notice until it stops working. Imagine woke up one day on a small boat in the middle of the sea storm. For people with balance disorders, that sensation can be part of everyday life. The utricle is a key organ of our balance system, constantly keeping us steady, oriented, and able to navigate the world. The utricle senses gravity and acceleration. And now let's step inside the inner ear to reveal the molecular composition of one of the boldest, smallest organ, hidden deep within one of its hardest and densest bone of the human body, the temporal bone. The inner ear houses the vestibular system, including the utricle, which is responsible for detecting head tilt and horizontal linear acceleration. The utricular sensory epithelium is characterized by 2 cell types, and they are mechanosensory cell types, type 1, which are flask-shaped, and type 2, which are cylindrical in shape. Both types possess stereocilia at their apical surface, and non-sensory supporting cells constitute the other major cell type. In this utricle cross-section, sensory hair cells are shown in green, supporting cells in magenta, and innervation in white. And they are revealed by specific markers. In this inset are visible 2 distinct hair cell morphology, type 1 and type 2 hair cell, which exhibit SOX2 positive magenta nuclei. That is a marker also expressed in supporting cell population. Motion detection relies on hair cells, which transduce mechanical motion into signal transmitted via their innervating neurons. Supporting cells surround hair cells and maintain tissue structure and ionic homeostasis. So vestibular hair cell acts as a movement detectors. Their stereocilia are embedded in a gelatinous membrane that contains calcium carbonate crystals called otoconia. When the head tilts, gravity pulls the otoconia, causing the gelatinous membrane to shift relative to the sensory epithelium. These displacements bend the stereocilia, triggering mechanotransduction and providing the brain with a rapid estimate of head motion. We are born with only 30,000 hair cells in each utricle, a remarkably small number compared with the more than 130 million photoreceptors in each retina. Vestibular disorders affect people of all ages, and more than 1/3 of adults over the age of 40 experience some form of vestibular dysfunctions. Despite a pressing clinical need, no pharmacological or biological treatments have yet been approved for vestibular disorders. These conditions are life-altering, and in response, the Vestibular Disorders Association launched the Balance Awareness Week more than 25 years ago, an annual initiative to increase public understanding of balance disorders. Its official mascot, the flamingo Fiona, symbolized the balance by standing on one leg. Clinically, symptoms such as dizziness and vertigo are a leading cause of falls, particularly among older adults, and are responsible for approximately 74% of traumatic brain injury in this population. Vestibular disorder represent one of the most costly disease categories in the world due to the aging population. And in the United States alone, the annual societal cost exceeds 150 billion. To begin addressing key questions in vestibular inner ear research, we secured a national grant, and we recently published a paper in Nature Communications revealing heterogeneity and damage response in the adult human utricle. Today, I will present some key findings and highlight a few ongoing projects in the lab. This graphical abstract summarizes our discovery. The adult utricle presents cellular heterogeneity and mounts a rapid transcriptional response after ototoxic injury. Using sequencing technique, we identified different non-sensory cell types, including previously uncharacterized supporting cell-like population. Following ototoxic damage, supporting cells activate early stress, remodeling, and proliferative gene programs within only 24 hours, highlighting the regenerative potential in the human balance organ. Sequencing technologies such as single-cell RNA-seq and bulk RNA sequencing detect which genes are present and therefore expressed in each individual cells within a specific organ. I will focus on three main learning objectives: cellular diversity, so the utricle is comprised of distinct supporting cell types with unique gene expression profiles. Damage responses, some supporting cells rapidly express genes involved in damage and tissue remodeling after injury, and clinical potential. These findings identify druggable targets that may guide future approaches to address vestibular disorders. One challenge to investigating the genes and pathways of human balance organs is basically the limited accessibility of these tissues. However, here at Sunnybrook Hospital in Toronto, we have the opportunity to collect adult utricle via translabyrinth surgery from people with vestibular schwannoma. In this image, you can see the size and the intactness of a utricle. We hypothesized that the adult human utricle has the capacity to respond to damage and initiate a regenerative response. To test this hypothesis, we set two aims. First, to examine adult human utricle sensory epithelium heterogeneity at single-cell level. Second, to investigate the utricular response to damage by tissue sequencing experiments and statistical analysis comparing control and damaged samples. The utricle, located in the vestibular portion of the bony labyrinth, contains a sensory epithelium with hair cells that are highly sensitive to damage caused by aging and ototoxic drug, such as aminoglycosides, antibiotics, and cisplatin. Once lost, vestibular hair cells do not regenerate, leading to progressive and irreversible balance disorders. In contrast, supporting cells survive hair cell loss and can generate new hair cells through proliferation and transdifferentiation, making them a promising target for regenerative strategies. However, in mammals, this regenerative capacity declines with age. In the human utricle, very little is known about the non-sensory support cells. Therefore, we performed single-cell RNA-seq of the sensory epithelium, and our analysis identified 8 distinct cell clusters, shown in a human plot, where each dot corresponded to a single cell. Cells that sharing the same color belong to the same clusters. And the closer the cluster are, the more similar they are. In addition to type 1 and type 2 hair cells, we found 6 transcriptionally distinct supporting cell types. These data are the first step towards discovering different supporting cells in the adult human utricle. We confirmed the presence of some of these cell population using RNA in situ hybridization, a technique that allows RNA molecules to be visualized directly in the tissue section. In these images, each dot represents a single RNA molecule. We use 3 cluster-specific genes, shown in green, for each supporting cell cluster, and demonstrated their expression in the utricular sensory epithelium, identified by SOX2 stained in magenta and cell nuclei in blue. The adult human utricle comprise distinct supporting cell types with unique gene expression profiles. To identify novel genes, we run a differential expression analysis between hair cell and supporting cells. This volcano plot illustrates the genes enriched in these cell types. A volcano plot is a graph that shows which genes change the most between 2 groups. Here, each dot represents a gene, and with the size of the change in the x-axis and the p-value on the y-axis. Several of these genes have not previously been described in human utricular cells. One example is SEGB2A1. This secretoglobin gene is specific to hair cell, as we can see in this UMAP feature plot. We validated its expression in the human utricle. As shown in these immunofluorescence images, secretoglobin protein is labeled in magenta, while hair cells are shown in green, marked by myosin 7A. In the merged channel, almost all hair cells express both proteins. In the supporting cell population, one distinct cluster is more separated from the others. To understand what makes this cluster unique, we compare its gene expression profile with the one for the other 5 supporting cell clusters. And this analysis identified specific marker genes, including ankyrin D1 and CRY-AB, that are highly enriched in this population, as indicated by the arrowheads. We therefore termed this group supporting cell-like cell, or SCLC. The exclusive expression of this gene in this population suggests that supporting cell-like cell represent the supporting cell subtypes with unique biological features. We used RNA-seq, RNA in situ hybridization to confirm the presence of supporting cell-like cells and their specific gene markers. Expression of ankyrin D1 in magenta and CRY-AB green was restricted to this cell population, as shown here, where each dot represents a single RNA molecule or transcript. Notably, supporting cell-like cells are located near regions of cell loss, indicated by an asterisk. In addition, supporting cell-like cells express genes associated with inflammatory responses and regulation of cell proliferation. Having characterized the utricular heterogeneity of the adult human utricle, we next applied a damage paradigm. To assess the immediate response of the adult human utricle to ototoxic injury, including potential early regenerative signals, we exposed the utricle to gentamicin-treated or controlled culture media for 24 hours and selectively damaged hair cells. We then isolated the sensory epithelium and performed either bulk or single-cell RNA-seq. Statistical analysis provides a cell-by-cell molecular characterization of hundreds of cells per sample, allowing us to define the earliest transcriptional responses to damage. Finally, we orthogonally validate selected target genes. The utricular organ is highly complex. We collect only the sensory epithelium and perform the bulk RNA-seq, and which provides an average gene expression profile across all cells in the sample. Bulk RNA-seq is like making a smoothie. All fruits, the cells, are blended together, giving a general flavor, but masking individual differences. In contrast, single-cell RNA-seq is like examining each fruit individually, allowing us to identify the unique characteristics, the transcriptome, of each cell population. This heat map illustrates differences in gene expression between 3 controls and 3 gentamicin treated utricles. In this plot, each column represents an individual sample, and each row represents a gene. Genes that are upregulated are shown in yellow, while downregulated genes are shown in magenta. Differential expression analysis identified 47 genes with significant transcriptional changes between control and damaged samples, including 5 transcription factors. Transcription factors are important because they are regulator of gene expression and can drive coordinated cellular responses to injury. In addition to bulk RNA-seq, which provides average gene expression, we perform single-cell RNA-seq to profile gene expression changes at cellular resolution in the same cohort of utricles. In these heat maps, it compares gene expression in control and damaged utricles. Column represents samples, control, and treated utricles, and rows correspond to more than 350 genes. Colors indicate gene expression, yellow for high and magenta for low, and treated samples show many genes downregulated here in the magenta plot. Among the downregulated genes, we identified transcription factors including NIFIB. Nuclear factor IB is implicated in retinal neuron regeneration in zebrafish, chicken, and mouse, and is downregulated immediately after injury. NIFIB regulates proliferation, differentiation, and cell survival. This violin plot shows its significant downregulation in gentamicin-treated utricles after 24 hours. Overall, this finding suggests that the adult human utricle can mount a rapid response to damage and potentially initiating regenerative processes. Among the supporting cells, we found a cluster that we renominate early responding supporting cells. These cells express unique genes like metalloproteinase 9 and EDNRB. This last one is has an homologue of is the homologue of chicken EDNRB2, and it was shown that it's an early marker for supporting cells to hair cell transdifferentiation in the avian cochlea, the hearing organ. Here in green, we show the validated expression of these genes in the utricular sensory epithelium, marked by SOX2 in magenta with nuclein staining in blue. Therefore, some supporting cells rapidly express genes involved in damage and tissue remodeling after injury. And likely some of these genes are responsible for new hair cell formation, similar to what other colleagues showed previously. Our transcriptomic analysis is the first RNA-seq study on human utricle that delineate the earliest response to ototoxic damage. In conclusion, we built a human utricle gene atlas at single-cell resolution and elucidated supporting cell heterogeneity. We discovered novel human cell type-specific genes, for example, secretoglobulin, and we demonstrated that the adult human utricle has the capacity to respond to damage and potentially initiate a regenerative response after gentamicin treatment. Our findings identified druggable targets that may guide future approaches to address vestibular disorders. We identified candidate targets that can be directly introduced or modulated into utricle explants, allowing us to study their effect. We are building a translational medicine platform using the utricle as an in vitro culture system model to test genes and drugs, enabling cost-effective evaluation of therapeutic strategies and accelerating a new era of inner ear regenerative medicine. Our vision is to advance precision medicine to treat vestibular disorders. Our adult data define the endpoint, the limited regeneration. To understand how this limitation arises, we next examined the fetal human utricle, which has a higher regenerative potential, as well as other fetal inner ear organs. I will briefly highlight a few ongoing projects as they offer potential opportunities for collaboration with modelers and theorists. In this study, led by Alice, a PhD student in the lab, we perform multi-omic analysis combining single nucleus RNA sequencing and ATAC sequencing to capture both transcriptomic and epigenomic information. We also applied spatial transcriptomics, revealing that the utricle is organized into distinct regions with unique transcriptional profiles. Using neighborhood analysis of the spatial data, which examined the local cellular environment of each cell, we mapped 3 major regions and identified region-specific markers for each cell types, including novel genes. We provide a high-resolution map of the developing human utricle and insight into human-specific mechanisms to inform strategies to ameliorate balance disorders. Then, we developed a decalcification method compatible with RNA preservation and applied spatial transcriptomics to fetal inner ear bone labyrinth sections from a gestational age of week 20. Case structures, including the cochlear sensory epithelium, the stria vascularis, and the spiral ganglion, were clearly distinguishable. Profiling was performed with 10x Genomics Xenium platform, and analysis of the tissue-specific genes is ongoing. We also profile these 3 main inner ear structures at both transcriptomic and epigenomic levels, and we are currently analyzing these datasets. We invite you to join us in exploring the human inner ear development and regeneration, and in particular, we are seeking collaboration with modelers and theoretical biologists, and we have an open postdoctoral position in bioinformatics or computational biology. I would like to conclude by thanking everyone who contributed to the success of this project. In particular, I would like to acknowledge my 2 lab mates, the sample donors, the surgeon, our collaborators, and the core facilities, as well as the funding agencies. Finally, thank you all for your attention, and I would be happy to take any questions.
In terms of development, and also that in terms of adult gene expression, what's the relation between the hair cells in the utricle and the hair cells in the cochlea? How similar or different are they, and how similar or different are their developmental processes and patterning in early development?
This is something that in the community is still an open question, because many people will say that type 1 is more similar to the inner hair cell in the cochlea, and type 2 in the utricle are more similar to the outer hair cell in the cochlea that are the amplifiers of the sound. But this was based on study that studied the innervation of these cell types and compared it. So molecularly speaking, there is, of course, gene in commons, but in the human utricle and cochlea, very little is known. And at the time aware, right now, there are no public data set available on human fetal inner ear organs in general. There are only some that studied the transcriptome of some inner ear fetal tissue, but didn't went in depth in the analysis of cell types and composition, and in particular transcriptome and epigenome, or spatial even. But I know some group in Europe and China are studying similar samples. So soon we will know more about them.
You say that you wanted to go a little bit more into modeling, and that typically requires having a sense of time. So what happens when things change? And typically, omics gives you snapshots. So you mentioned something about in vitro, and I think that that might be a way to go into the time, but I wanted to ask your view on that, or in general, of course, on this topic.
So thanks, Lorenzo, for the question. I think that having a modeler or someone that can help us to model the response when we, for example, create damage with a drug, an aminoglycoside, we have 2 time points in terms of time points that you were asking. One, that it's more when the cochlea, for example, do have the capacity to regenerate, that it's around week 15 gestational age. And one, it's when the cochlea lose this capacity, that it's around week 20, 18, 20. And so there we can understand the differences between stages. But to understand which transcription factors or cocktail of transcription factors we can use in week 20 to induce the regeneration that we see in week 15, we need some more powerful analysis to try the combination of all these transcription factors and regulators we found from our dataseq, because it's unfeasible for us to try thousands of combination of transcription factor that can allow the feature, for example, creating the bundle in the surface of the sensory hair cell to after be functional, while modulating these in silico can be a faster approach of which transcription factor are the core transcription factor, the Yamanaka factor of the inner ear to regenerate after the organ and make the function in vitro. So we can go directly in vitro to test the few combination and try to accelerate this process.
Okay, so you're saying that perhaps we need a little bit more of bioinformatics to really pin down to those genes that are really key and then move forward with those in a more feasible approach.
Yeah, no, I was going to say, I mean, obviously it's clear that there's a bioinformatics, but maybe you can, also discuss, like, there's a potential for theory to be tested in a different way. I think we've seen a few talks where you can, put a network and try to explore how these genes influence one another. So I just want somebody who's not doing bioinformatics, but has a potential to test hypothesis or also show you where maybe how things change or damage. And then maybe also test how these drugs or what are the potential drugs that you can target, right? So there's a lot more potential than just bioinformatics analysis, but we can push it further, right?
Yeah, I agree. There is very little known in the inner ear, although the organ of Corti, the sensory organ for hearing, was discovered over 150 years ago. So there is very little known because it's very hard to access this tissue. They are super small and fragile every time that we try to do sequencing, or, for example, the calcification for the whole temporal bone can damage the RNA quality. And so this hampered the investigation of molecular targets and future therapy for the inner ear disorders, hearing or balance. So I agree that having biologist theorists that can help us to understand how can be exploited these data sets that are very high quality and taking multiple time points to get back to Lorenzo's question. So we have at least 2-3 time points per organ. And so we can understand how development happens, but also when we treat with a damage paradigm, when we apply damage paradigm, what is the perturbation of the genes that can be modulated. So perturbating in silico these genes, transcription factors, how we can modulate in silico to after streamline the process in vitro.
Yeah, I'll make another comment. I just want to say there's nothing nicer than the unknown for modelers, right? So that way we can show what's in the black box of development, but we could use the white mechanistic, white box mechanisms, and maybe we can even target some mechanisms. So I'm just opening this up for anybody who have ideas or would like to be part of this, that it's not restricted. And the more unknown, the more the in silico is going to be useful. So
thank you. I will look forward to receiving your email or comments after this meeting.
I want to follow up a little bit on Lorenzo's comment, and I might disagree a little with Maria. You have to know a lot to be able to build models, and you have to ask the right questions. You have been doing spatial correlation analysis. And so you're trying to understand niche organization and structural organization of the tissue. That typically is very important for building models that are effective. We know that in cochlear development, the formation of the hair cells is extremely patterned. The position of individual hair cells is determined with very high precision in early development. Do you know if that's true in the utricle as well?
Wow, what a question. Yes, it's so we know that utricle is very different in terms of developing hair cells. So as you just mentioned, the cochlea is tonotopic. So the formation of the cells, hair cell in the cochlea happen to inner versus outer and the base to apex. In the utricle, there is no such pattern, but definitely there is some things that guide the formation of these hair cells, because we see these hair cells popping up from the striatal region, extrastriatal region. So there is no that gradient that we see so clear in the cochlea, and this makes the development of the utricle even harder to study. So having the transcriptional transcriptomic of spatial transcriptomic of the utricle can help us to guide this time point of development, and we so far have collected 2 time points, but we are hoping to add more to have this trajectory. What we are seeing that these hair cells are showing very early in the development around week 12 across all the tissue, but the proliferation of supporting cell that after became hair cell decline around week 18 already. So we saw that the proliferative clusters in the utricle diminishes and also doesn't have the pattern that has the cochlea. But a sensory cell to be in the specific site there has to be signaled from other cells around that say, hey, I need another type 1 here close to me because I have only a type 2 here. So for sure there is some signal going on to decide where and how the new cell form in the striatal, extrastriatal region. But we don't know yet what guide this. As the cochlea, it's a very clear, but in the utricle, it's not super defining.
So you mentioned, and Lorenzo asked about this, that you're doing explants. So you have the tissue, you have postoperative tissue, and then you're putting it, and you said that you're doing a systemic gentamicin damage and then looking at 24-hour post-injury. How long can you keep the explants going? How long could you observe? Could you observe 2 days, a week? How long could you possibly observe?
For now, I was only able to culture utricle until 21 days, adult utricle. And I did the same for the fetal utricles, but we would like to exploit more this with organoid systems. So we dissociate the tissue and generate like custom organoids, let's say, to understand if we can culture for a longer time. And so go over the 3 weeks for the whole organ, because more the organ is in culture, they become curly, so it curl the surface, and after it's not so accessible to do IF or immunohistochemistry or things like that. So we would like to shift in the organoid sides that help us to resemble the tissue and have more replicates to test drugs or compounds, because when I culture in the whole dish, it's 1 utricle per dish. So I can only do 1 experiment at a time. And after, if I split in multiple mini organs, I can modulate better time point, drugs, and genes.
But if you can keep it going 3 weeks, that gives you plenty of time to observe the pattern of proliferation. It gives you time to look at transdifferentiation.
We noticed that a utricle is slow. Maybe because it's human, maybe because it was adult, but nothing happened so quickly like the mouse or the zebrafish. And these require probably more time. So we were planning to culture for like over 2-3 months. But so far, these experiments are ongoing, and we'll see how the PhD students will take on these projects.
So could you clarify one other thing? You mentioned you don't get direct proliferation of the hair cells, but you said that even in adults, there's some limited degree of transdifferentiation of supporting cells back into hair cells after damage.
Thanks for making this point because it's very important to clarify. So the cochlea lose this capacity, while the utricle has retained some capacity even in adult stage. And that I was referring to some hair cell, as you saw the small nascent hair cell in scanning electron microscopy that I did. You can see it's resembling a hair cell, but it's smaller, the bundle are shorter, and it's a new formed hair cells. But how this hair cell is functional and how many hair cells in adulthood we need to recover the function, that is not unknown, because so far we saw this process. So there is the capacity, but we need some factors to increase until we restore the function. So the utricle, yes, retain this capacity. The cochlea completely lose this capacity after week 20 of gestational age. So as soon as we're born, we start to lose hair cells. I mean, maybe I was more dramatic, but sounds and things, we need to protect the hair across all life because we only have few thousand of hair cells.
Are there existing in vitro proxy that work, or cell lines that one can use for simpler experiment to really tune the nuts and bolts of the potential model?
Is what the field lacks. We do not have a human cell line for balance organ or hearing organ. So we use some kind of pluripotent stem cells and things like that, or from mouse that are immortalized, but there is nothing that really is a primary cell line that after was immortalized and really fully resemble the human organ. We are working on this, but as you can imagine, there is many regulatory and ethical concern using fetal tissue for doing so. So the community for now is moving more for iPS and pluripotent stem cells than generate a cell line from direct tissue, like the fetal tissue, and immortalize the cell line.
Are the protocol for IPSID already established, or again, is one thing that we need to?
It's established. There are a few groups in US and Europe that establish very meaningful pipeline that give you results and resemble the hair cell and supporting cells transcriptome as well. So it's very similar and can be used for modeling drugs and genes for sure. And already there are publications in very high impact journals that show the potential of these cell lines.
So you mentioned the difference between development and function of these organs in rodents and in human. How well mapped are they, especially the early development and recovery after damage questions, in mice or rats, for example? And how much homology do you see in the gene expression patterns in the animal models compared to the human?
There is a huge difference there. There are many gene in commons. When I did the cross-species comparison, for example, of the utricle in mouse versus human, there are many gene in commons, but some are unique, like the secretoglobin. It's a unique specific gene on the human hair cells. But the map, to come back to your question about the comparison between mouse and human, there is a very good paper published by a colleague in Europe, Marta Roccio, and she showed that, for example, P0 mouse resemble a weak 15 cochlea. That is where the regenerative capacity is still ongoing. And week 18, week 20, it's around the P8, so where the mouse also lost the ability to regenerate the cochlea. So there is a correspondence, but clear match, a one-to-one match, it's not well defined. And we are still currently doing this analysis to understand which is the window exactly in mouse that is P0 in human, that correspond to mouse P0, P5, regenerative, non-regenerative cochlea. P stands for postnatal day after born. So we know that the window is week 15, week 18, 20 for the experiment that we have done in the lab, but we are still under evaluating this to see and define the precise window. So we are working on that to match the exact temporal stage between the two species.
I find it very interesting because I know quite a few people who work in detail on computational modeling of cochlear development. And so I'm trying to get a sense of how much that might or might not translate into models in another organ system that's related. So you showed us some pictures. What's the typical hair cell spacing in the adult? Is it one cell? Are they basically deads? Is it a dense epithelium with a substrate? You showed quickly a slide showing the stratified. I think it is a stratified epithelium architecture. And you said that two hair cell types are paired typically. But I'm trying to understand what leads to the specific structural architecture of the tissue. What defines the hair cell spacing? What defines the spacing between the various supporting cell types? Because understanding those spatial relationships, as you pointed out, is critical to understanding the development and maintenance and potential recovery of the tissues.
The epithelium is formed by basically, if we want to define, two main cell types. Non-sensory supporting cells that surround the sensory hair cell that captures sound in the cochlea and movement in the balance organ, and the sensory cells. Now, there are multiple subpopulation of supporting cells in the cochlea, and less defined population in the Utricle. So we were one of the first group to characterize this diversity. So we know that there is a diverse compositions, but it's not easy to answer your question because there are still so much project to dig in to answer this question, because I don't know, and we don't really know how it's organized and why a type 2 it's right there instead of a type 1. And we know that adults, Utricle, lost more type 2 compared to type 1. But type 2 are the ones that are more easily to regenerate compared to the type 1. And so who knows how it's related to regeneration components, but also in the development, who define how many type 1 and type 2 we have, or where exactly they are. So there are so many questions that we would like to answer in the community. And some of those, there are groups that start to answer this question, but I need to be honest, I'm not 100% sure where they are now.
Well, for example, in many organ systems, when you have this kind of distribution, both the initial structuring and then later stem cell niche maintenance are carried out by delta notch juxtacrine signaling. Is that a critical factor here in the distribution of hair cells versus support cells, either early on or later in homeostasis?
Yeah, notch signaling play a major role also in the hearing and the balance organs. So yes, there is that component, but there is also wind signaling component that play a major role in the formation and development of the cochlea as well, and in the Utricle too, ampullae, like the other sensory organs, balance organs. So there are these common pathways in other organs that are also expressed or active in the inner ear. I think it's also important not just to focus to what it's overexpressed in the cochlea during development, but what it's turned down during development and why that are down-regulated to have that kind of composition of hair cell and supporting cell properly done and organized spatially and tonotopic for the cochlea, for example, during the development. So I want to dig also in the downregulated transcription factor and genes to understand, because probably there is some epigenome components in the adulthood that maintain that gene silent, and we want to access the chromatin and start the expression of the genes. So it will be very interesting to study both and understand how these can be modulated and probably tested in vitro with these explants that we receive from the biobank.
Well, I think 3 weeks is probably long enough to study a lot of the interaction mechanisms. I mean, when I look at trying to understand a system of the kind you're talking about, what I want to know are what cells are sending signals, what cells are receiving signals, how are those signals mediated, how long do they take for the signals to transmit, and then what is the response to the signal when it's received. And I think in your in vitro systems, your explant systems, you could probably map a lot of those things. Probably, I think you said your gentamicin treatment is basically systemic. You're not doing a focal damage, or are you doing focal damage?
No, it's the whole utricle in the media treated with gentamicin and control. Yes, it's the whole organ being damaged.
So to study signaling, it might be productive as well as doing those sort of systemic damages to try, for example, to do laser ablation or some kind of focal damage, and then look at the spatial gradients after damage to try to get a sense of how signal transduction is happening during regeneration, because regeneration certainly depends on what happens inside of individual cells, but it's almost never a phenomenon purely of a single cell. It depends on the ensemble of cells and how they interact. And it seems from the pictures that you're showing that you have a very impressive ability to analyze those kind of spatial and temporal interaction structures. And so that potentially could be something I think might be worth thinking about.
We are working on this. We didn't do the test in vitro yet, but we did some bioinformatical analysis using a tool called CellChat to understand the ligand receptor connection and which population talk, as you just mentioned, communicate with which one, or which signal are sent back and forth to some specific subpopulation to make a week 15, that is the regenerative cochlea, the communication ongoing, versus week 20. There is a reduction of signal. There is some change in the communication between the populations there. So what are the main ligand receptor and pathway of those that are implicated in this communication? And so if a cochlea do not regenerate anymore, which are the factors that stop this communication? Or so we are working on this, and thanks for the question, because I think it's worth to also expand in vitro and try to understand after culture how we can make a time point of this communication better during the culturing. But for now, we are just doing this at 2 gestational ages and comparing these differences in transduction communication processes.
Lorenzo makes the point in the chat that, of course, when you're using human explants, those are precious. You don't have so many, so it's not such an easy experiment. But doing titrated low-dose perturbations is often more informative than high-dose cytotoxic perturbation. If you do low-dose perturbation and do titration on that, assuming you have enough samples that you can do that, then you often get more informative dose response curves that can then help build predictive models more easily. If you're mapping primarily the architecture of interaction, then the single hitting it hard and seeing what happens is always the place to start. But I think Lorenzo's point about trying to look at dose response for injury is an interesting one.
We are trying to, in another project, we are trying to characterize these responses. So we started with a very low dose, and we didn't saw any effect, visually speaking, like caspase-3, that it's one of the markers for apoptosis and damage, being overexpressed expressed in the hair cell. So we start to increase the dose, and we now are using a crazy amount of aminoglycoside in the explant, in the fetus explant, to see an effect. And this is telling us the resistance that these few thousand of cells we have in the organ have on these aminoglycosides. Although it's an in vitro experiment, we arrive at like 20 millimolar of these drugs, and we now are seeing some effect after 1 day in culture with the drug, and after 48 hours of recovery after the damage. And we now see some hair cell being damaged, so some caspase-3 positive hair cells, so it means we really are killing them. So now that we collected this data set, we can start to sequence them, and these experiments are really fresh. We did this week, and so maybe next week I will start to collect the data set, and probably we need an earlier time point, like a lower dose, but at 10 millimolar we couldn't see any effect, so maybe we should try 15 or see to culture longer. Like instead of only 48 hours of recovery, we could do a week to see if we kill, and after some things, new hair cells are arising.
I mean, something we found working in the liver, for example, after toxicant damage, is there are a lot of early responses. Actually, a lot happens before 24 hours, at 2 hours, at 4 hours, at 8 hours. We were in rodent, but the liver is similar in human. But there are these rapid responses, not the long-term recovery, but the early rapid responses can be quite determinative of the long-term outcomes. And so, again, I understand the complexity of trying to do these kinds of experiments when you have limited supply of samples and their endpoint measurements and their expensive measurements.
Human variability.
Yes, right. And your individuals aren't directly comparable. So I'm not trying to say that it's an easy experiment to do,
No.
but in a sense, that's why I was asking about the animal models, because you might be able to rough out some of your understanding of some of these processes in mouse, for example.
Yeah, this was already done.
And then refine it in human.
Yes, this was already done and published in the inner ear. So we are using these people that already have proved how damage works to tweak and do in the human, but it's still a very not trivial process.
Yes, you have beautiful data. It's really an impressive set of experiments. No, I want to thank you very much for presenting such an intriguing talk. I'm looking forward to hearing follow-up on how your data sets develop. If you could come up with an organoid model, for example, that would be really exciting. And I hope that you'll find collaborators who have the modeling of the signaling and gene regulatory processes that can help you build predictive models of the system. It's definitely one that's worth pursuing. And I think that the community has the ability to build models of the kind you're looking for a system of this kind. And so I hope that we can help you make the connections to do that.
So thank you.
Okay, well, I want to thank everybody again for coming today. Remind you that we don't have a steering committee meeting tomorrow, and we'll see you at the next seminar. Thank you so much.